Welcome to Innovation Out of Failure, where we talk to people about their careers and, most importantly, about the challenges they have faced that actually end up making them more successful innovators in the end. I am Andrew Lyon, Dean of the Fowler School of Engineering at Chapman University, and I'm your host for this series. This week, we're speaking with Professor Michelle Dawson. Michelle is a bioengineer on the faculty at Brown University, where she leads a research group that is laser focused on obtaining a deep understanding of cancer and how to develop engineering approaches for its treatment. All right. Welcome to Innovation Out of Failure. Today I am joined by Professor Michelle Dawson from Brown University. Michelle, thanks for joining us today.
Thanks for inviting me.
So you, like me, are an academic. I wanted to get a little bit into kind of your background, what you're doing right now, and really how what you've done previously informs your research today. You've got a really interesting background from an educational point of view, biomedical engineering, chemical engineering, and even a stint doing radiation oncology. Maybe you can tell us a little bit about how that winding path took you to where you are today as a researcher and a scholar.
So, it's like, I guess I've kind of followed the path that I was led on with each decision. And, each decision I looked at independently, but my goal in the end was always to do cancer research. And so I kind of took a tortuous path to that process of getting the background I needed to really understand cancer and to be able to study it at a level that would be clinically relevant. And I think that the education I have has, in some ways, been more tortuous of a path than it needed to be, but at the same time, it's helped me to have a unique kind of perspective in looking at cancer and how we can develop strategies for really altering its path. And, I started out in biomedical engineering because actually I wanted to be a biologist and I really loved biology, but I was also, really enjoying math and enjoying kind of the logic between the engineering classes. And so biomedical engineering was a good way to kind of combine my love of biology and of kind of logic and math to be able to come up with a degree that kind of captured all the things that really interested me. And then when I moved to Johns Hopkins, I felt like biomedical engineering was great because it allowed me to explore all these different areas, but that it didn't have the focus that chemical engineering had. And I had always been interested in kind of a cell and how a cell functions as a reactor. And that cell as a reactor model is something that's used in synthetic biology, it's something that's used in engineering cells as therapeutics, and in many other systems. And so I thought, learning kind of this chemical engineering discipline would help me to understand a cell at that level. And so that was one of the driving factors that led me to chemical engineering. And going to Johns Hopkins, when I met Justin and heard more about his research, which was focused on drug delivery and designing particles that could move through biological barriers to treat disease, I thought, wow, this is really interesting. And being able to design systems that could interact with the body that way would be really, clinically relevant, and it would motivate me. And, this is something that I wanted to do. And so I decided to take that path. But at the time, he was really interested in pulmonary drug delivery. And I was working with a spray dryer, and I was spray drying particles to make these large light particles that could deposit in the deep airways. And it was the worst graduate project that I could imagine. It was not fun at all. And so I spent a lot of time reading and thinking about what the biological barriers were to drug delivery in the lung. And the barrier that kept coming up was mucus, because mucus coats the majority of the airways, and mucus is designed to remove things from your airways. And so if you are a drug delivery particle and you deposit it in the lungs and you don't make it to the deep airways, then the likelihood is you will be swept out of the lungs using this mucociliary transport. And so to design things that would adhere to mucus or move through mucus more effectively is one way to increase the drug delivery rate in the lung. And so that was what I started to think about a lot. And I told Justin about my idea, of looking at kind of drug delivery through mucus covered surfaces, and he really wasn't that interested in working on it. And so he said, if you get a fellowship, then you can work on it. And so I got the NSF fellowship, and then I got the Ford fellowship. And so he said, fine, you can work on it. And so I started working on that project, and within a year, I won the ISAM award, which is the International Society in Aerosol and Medicines award, because this was something people hadn't been doing. They hadn't been using particle tracking to look at how drug delivery particles move in mucus. And this is one way to be able to test the surface and test the size of a particle to make sure that it can penetrate these barriers in the lung. Mm-hmm. And so this really kind of changed things for a lot of people in the field. And so that was one way that I definitely innovated out of failure, was to come up with this idea that led me to this new project. And one thing that I learned in doing this project was that it was really fun to use biophysics and to understand biophysics and to have a biophysical model that you could mathematically develop that would tell you information about a system, and then to have experimental data and be able to combine them and understand how empirically this data kind of matches this biophysical model and where it deviates, and how you could learn things about those differences through more research and through increased understanding of your system. And so that was something that I think, I carried with me from that research experience. And but I really wanted to work on cancer, and that was something that had always motivated me. So I decided to make this really big change and move to Mass General, working with Rakesh Jain, and looking at cells and how they move within biological systems in mice. And so when I was in Rakesh's lab, I did bone marrow transplant on mice, and I looked at how bone marrow-derived cells move in the body in response to the development of cancers. And so we did things like IP injection of tumor conditioned media to kind of prime the tissues to see whether that would increase the level of bone marrow derived cells that accumulate in certain tissues prior to the arrival of cancer cells. And so I spent a lot of time, doing bone marrow transplants on mice and trying to understand how these bone marrow derived cells move in the body in response to the development of cancers. And that, was really interesting. I spent a lot of time in the lab and just really immersed myself in this field. And being at MGH was a very different experience than being at Hopkins, because I was really at the hospital and I was kind of living, around a lot of people that really understood cancer and used acronyms to describe everything. And so immediately I had to learn the language. And, I spent time doing that and developing all these new research tools, which were completely different than what I had been working on before. And it was kind of like having a whole nother graduate degree by the time I was done. And so after this, I really needed to find a faculty position. I was at a kind of critical point in my life because I had a daughter who was about ready to start high school. And so I knew that it was the time that I needed to make the move. It wasn't the perfect time in my projects, but I had to move at that point. And so I took my first position at Georgia Tech, and I went back into chemical engineering, which, is a good place in some, in some universities for somebody working in cancer, and others maybe less so. And so, it was not necessarily the best decision again for me, but, I've gone from there now to being back in science and biology. And so now I'm at Brown, and I'm in molecular pharmacology, physiology, and biotechnology. And when I was at Georgia Tech, I spent a lot of time kind of developing mathematical models that would help us to understand the data we were collecting on how cells move and how their mechanical properties change in tumor-like environments. Here at Brown, I'm really in a space where I can interact with the clinicians on a regular basis. I have a lot of clinical collaborations where we get tissue from the hospital, and we're able to analyze those samples using our tools. And so it's really, allowed the lab to kind of develop in a more clinically relevant path. Mm-hmm. And so My focus lately has been on chemoresistance and what kind of cells develop chemoresistance. We've been very focused on heterogeneity in cell populations and how the cell fate processes change in response to the development of cancer and drug delivery.
So thank you for sharing that. I mean, that really is a fantastic lesson, I think, in nimbleness, right? Being able to pivot from place to place, and the field you're in right now, I think, is also one that requires that kind of nimbleness, because it is so incredibly complex, the world that you study, and there's so much going on. I mean, it's a really dynamic field, and so you use the word innovation, but in our field, in kind of biomedical engineering type world writ large, that's an incredibly overused term, and so given the fact that it's incredibly overused and almost trivialized in many ways, how do you think about that when you talk to your students, for example, and you talk about really generating new innovations in this incredibly complex field? What does that mean to you?
Well, it has multiple meanings. Innovation can be, because really when we think about innovation, we think about looking outside of the normal and trying to find kind of a creative thing or something that's new or unestablished that you're going to add. And the first thing you have to do is you have to know what's there. And so I always tell my students that they really need to read a lot of papers and they need to understand the literature around what they're studying. I send my students papers all the time. We get tables of contents from all the major journals so that we can constantly update kind of our thinking in the field. Because what is new today is not going to be new tomorrow. And so you could be working on something and you may have to pivot if you want to be able to have, a top publication, even, if it's new while you're working on it. And so I think that innovation is like a moving target, and sometimes we know when we come up with something that's truly innovative. Like I have a student who's working on he's looking at the biophysical properties of polyploidal giant cancer cells. And when he identified these cells in his populations, people kind of thought that we were silly focusing on these giant cells. The pathologists thought of them as dead-end cells, and a lot of people thought, well, who cares? These cells are no longer dividing. But there was a paper that came out that talked about how they undergo amitotic budding. And so now you have this cancer cell that gives rise to other cancer cells through a non-mitotic process. And this leads to, advances in the disease because it can also alter the genetic makeup of those cells because of the way that this cell accumulates DNA from different cells that could then be kind of swapped out. And so it's a pretty important discovery to be able to study these cells. And so that was one of the things that I would say that we've done that has been really innovative over the last few years, is to really start focusing on these cells and understanding the biophysics of these cells, which hasn't been looked at before our studies. And so to me, that was innovative, but we weren't the first ones to discover these cells. These cells have been looked at by others. Someone else identified the fact that they undergo amitotic budding. There are other people studying them. But there's a handful of people studying these cells, whereas there's hundreds, thousands of people studying breast cancer cells in general. And so I guess innovation is doing something new that no one has done, but it's also doing something that is newer or that is kind of going against the norms so that you can come up with an idea that will help you to kind of bridge the gap in those areas, because we need detailed studies in addition to new studies. And so I think innovation is somewhere kind of between brand new and more detailed.
And so, okay, fine, lots of people are studying these types of cells, but certainly not as many as in the cancer world as a whole. Uh, but, is it the fact that you've entered so many different fields as an outsider that also gives you that kind of ability to look at that system and just look at it slightly differently, that allows you to then tackle it in a way that no one's really tackled it, or has at least perceived the importance of that in a way that lots of other people haven't?
Absolutely. Our interest in mechanics and understanding biophysics of cells really helped us to come up with something that was new to learn about these cells, because we were able to apply our biophysical approaches and our single cell approach to look at heterogeneity in these populations in a way that hadn't been done before. And so I think that is what made it more innovative than just looking at kind of a rare population of cells and only a few people were studying them. And so that definitely added to the field something that was new.
Yeah. So you've also already talked a little bit about failure. You talked about the, conflict that was constructive, a constructive conflict you had with Justin Haynes at Johns Hopkins, and you described how that failure really allowed you, or maybe in some ways forced you to go and strike out on your own, to demonstrate the importance of an idea. Is that really the most important failure in your career, or are there other things that have also similarly driven you forward?
Well, there's been a lot of failures, honestly. I mean, every time you get the response that your grant is not funded, or that you're not going to get a promotion, or that your experiment failed, or an idea that you had didn't quite pan out, they all feel like a little bit of a failure, some of them larger than others for certain. But I think, the biggest failure has been the things that I haven't done while I've been doing all this stuff, because I took this tortuous path. I spent a lot of time, trying to learn new things and immersing myself in these different cultures. And so I gave up a lot of family time. I gave up a lot of time with my daughters, and I can't get that back. And so now
Mhm.
it's like I wonder whether the decision to take such a tortuous path was a good one. I'm happy with, all the things that I've learned along the way, and, all of the skills that I've been able to accumulate. But at the same time, is there a better way to get to the end goal that doesn't require so many sacrifices? And that is something I think that people should think about. When you're deciding what you want to do, really trust your intuition and think, like, in the end, what do I see myself doing? Do I see myself working with mucus and understanding, particle transport in mucus? Is this the thing that motivates me, that I want to spend the rest of my life doing? If I had asked myself that question before, I would have known that the answer was no, and, I would have probably picked a different project. But I was interested in the things I was doing at that time, and I was kind of in the minutia and trying to
Mhm.
understand things the way graduate students do. But think about your long-term goals. Don't just live in the short time, but live in the long term, and think about how what you're doing now fits into your ultimate career goal. And so, kind of looking to the future, I think, is something that I failed to do early on, that I could have spent more time doing, and that would have helped me to make better choices.
So, is there also some of this thinking about internal versus external motivations, right? I mean, so I feel like I remember this being true for myself when I was a graduate student. Most of the motivations seem to be very, very external. And if I really reflect upon my time, there was very little kind of, I was certainly interested in some of the things I was doing, but the internal drive was really being chewed on and churned on by that external pressure that's being applied to you as a graduate student. Is there some of that, some notion that maybe we as students and researchers need to be more true to ourselves in terms of what we really want and what really drives us?
Absolutely. I think, when you have a student who's likely to go to graduate school and you see that spark, um, that's, the kind of sign that maybe that student, is really going to have that internal motivation. I had a student like that at Georgia Tech, and he's now a postdoc at why can't I think of the name, at MD Anderson, and he's done very well for himself. And from the moment I met him, he was a freshman, he had the spark, and I could see the spark. And so, that's something that you don't want to lose track of. Even though you have a lot of external pressure, I think it's important to maintain that excitement. And, when I was at Hopkins, I think the person who I saw that in the most was Denny Wirtz. He just runs around and he's constantly excited about everything. You could tell him about, how the particle transporting mucus is following this weird pattern, and he would get excited about it. And it's like there's so many things going on, but to be interested in this one thing because of some biophysical trend or something is something that he was capable of, because he had that deep interest in the experimental work. And that's something that I think is difficult to maintain throughout our careers. And so to be able to focus on things where you always can kind of feel that drive a little bit, I think is important.
I actually, when we started chatting, I didn't anticipate getting to this point, but maybe I could ask you your opinion on something, since we're hovering in this world of how graduate research happens. What's your perspective on kind of the future of graduate education in science and engineering, right? As we study more and more complex systems and things become more interdisciplinary, and the sparseness of funding becomes a real challenge for so many researchers. Do you have a perspective on where we need to be heading with graduate education and how it intersects with high-level research?
Yeah, so, when we were graduate students, probably most graduate students did a lot of wet lab work and spent a lot of time in the lab doing and designing experiments. Those experiments are really expensive, and a lot of the data that you collect with those experiments is underutilized. And so I think a lot of the new directions are going to be more computational. There's going to be a lot more databases that are generating data sets that people can analyze using computational methods. And so more students will probably follow a path that utilizes those resources to minimize their need for additional experiments. And probably, we're going to move from seeing 10 to 25% of students doing computational work to more than half of students doing computational work. Especially with the COVID-19 situation, we have students right now that are doing nothing but computational work. And, this is something I think that the university would like to see.
Mm-hmm, mm-hmm. Well, I'm going to say right now that I didn't prompt you for that answer. That was not a planted answer, but given that probably more than 50% of the students in the class are computer science students, I'm pretty sure that that notion and that future is going to resonate well with them. We're about out of time. Do you have any final thoughts you'd like to share with the students?
I'm just looking at my notes to make sure I didn't miss anything. And so I guess I would just say do something that you're passionate about. If you, see yourself working on a specific project, then follow, your intuition and try to get to that goal, and don't compromise your ideas. And, if you do something you're passionate about, then you'll do something that you will do well, and that's important. And so that would be my final thought.
Thank you. I think that's hits the nail on the head. Michelle, I'm really glad you decided to join us. Thank you so much for accepting the invitation, and best of luck to you in your work.
Okay, thank you very much.
I'd like to thank Michelle for taking the time to join us and share her perspective, and also thanks to you for listening to Innovation Out of Failure. I hope you enjoyed this episode, and I hope you'll check out all of my other interviews with amazing innovators. Once again, my name is Andrew Lyon. Thank you for listening.